Hi, and welcome to this webinar on the PGT-AI platform for analysing embryos for PGT-A. So I'm David Crimes. I'm the Business Development Director in the Cooper Genomics part of the Cooper Fertility and Genomics company. And so here today, I'll be approaching PGT-A through a molecular biology perspective. I'm not an embryologist, just to make that doubly clear. I'm a molecular biologist, so please take that into account when listening to this webinar. So Cooper Genomics contains within it over 27 years experience, including the pioneers and founders of PGT, Santi Monet and Marcus Hughes, both still associated and working within Cooper. So we've got lots and lots of experience. We've done now over 700,000 embryos, we think, that we've biopsied and provided results for through our accredited facilities. And in the UK, we have an ISO 15189 accredited lab for PGT-A, M and SR. And today we're really talking about the advancement in PGT, and that's PGT-AI. Specifically today, we're really talking about PGT-A, touch on SR a little bit, the structural rearrangements, but really PGT-A. And just as a recap, PGT-A is the testing for aneuploidy. Now, this has previously been known as PGS, pre-implantation genetic screening, CCC, comprehensive chromosomal counting. Some places even use the acronym PGT-A, pre-implantation diagnostic for aneuploidy, even though it was never a diagnostic test. It's always been a screening test. And it screens for chromosomal abnormalities. And it's an option for all IVF patients. It doesn't have to be offered to all IVF patients, but there's nothing stopping it being offered to any IVF patients. It improves IVF success rates. But the key and most important thing that separates it from the other tests is it does not require any personalized test preparation. As I say, that's PGT-A. This technology, PGT-AI, is also the technology we use for our PGT-SR, the structural rearrangement. This was previously part of the PGD wing, the pre-implantation genetic diagnosis. And this screens for the inherited chromosome rearrangements. But the key thing between PGT-A and PGT-SR is they're both counting the total number of chromosomes and the chromosomal content of the embryo. So they're a chromosomal counting test in both cases. Now, the process of running PGT, and for this particular one we've highlighted PGT-A, as that's the major use of this technology, is the IVF is performed in the clinic. Now, just to re-emphasize, for Cooper Genomics, we do not require ICSI. Some other suppliers do. However, we have shown conclusively that the technology we use is unable to amplify or analyze the DNA directly from sperm. So this is you can use standard IVF procedures for PGT-A and SR within Cooper. So after the IVF, you will carefully do an embryo biopsy and take 5 cells from that trophectoderm biopsy. After the biopsy is taken and you're sure that you've tubed the cells carefully, that embryo is vitrified, send it to our laboratory, we produce the results, they're released to the IVF center, and the IVF center, in combination with the patient decision, transfers hopefully a chromosomally normal embryo with the additional embryos remaining for future transfer. So the whole point of PGT-A is to screen to find these chromosomally normal or euploid embryos. So as a very quick recap on PGT-A, a euploid embryo is an embryo which contains the correct number of chromosomes, and we know that they have the highest likelihood of a successful pregnancy. So here we have the normal chromosome numbers, so there's 22 autosome chromosome pairs shared. So both males and females have chromosome pairs 1 to 22. If you're female, you're XX, and if you're male, you're XY. The key thing is you have 23 pairs of chromosomes, 46 chromosomes in total, which contain all the genetic information. Aneuploid embryos are embryos which contain an incorrect number of chromosomes. In this case, we've highlighted chromosome 10. No live birth has ever been recorded with a trisomy 10. So the whole purpose of PGT-A is to screen out embryos such as this, because we know that an embryo which is aneuploid may result in a failed implantation, which is the most likely outcome, a miscarriage, or in some cases, particularly with an SR case, genetic disease. So really, we know that euploids work best and aneuploids basically don't work at all. However, there's this third category, which is increasingly talked about, which are these mosaic embryos. Now, mosaic are embryos which have a mixture of both euploid and aneuploid. And we know from the data that mosaic embryos implant less, miscarry more, although can sometimes lead to live birth. So really, we're looking to really differentiate, and this is where PGTI comes in, and we'll look at some more data a bit later, is it's absolutely key to stratify to allow the best IVF choice between the euploids, the mosaics, and the aneuploids. And we will even go further than that and follow the PGT-AS guidelines to state that mosaicism should be split into two levels. So you have your euploid, which is your normal with the highest likelihood of success. We then have at the other end of the scale the aneuploid. They're completely abnormal, very, very low, very unlikely to produce any form of sustained pregnancy or live birth. And then sat in the middle, we have these mosaics. Low-level mosaic, which have a mixed outcome. It's lower than a euploid, but it's reasonable. And high-level mosaic, which basically have a worse outcome. They have a higher miscarriage rate. So really, we're stratifying these results into 4 categories. And this is really where we're coming down to making sure that when we say something is euploid, it is truly euploid. If we're saying it's mosaic, it's truly mosaic. And why does it matter? Surely, you know, as long as it's reasonably accurate, that's good enough. Well, actually, no. So you can find out more details about what I'm about to show here on mosaic transfers in the full webinar on mosaicism and all you need to know. But top level, just to remind everybody, when you transfer a mosaic embryo, depending on the level of mosaicism, so even low-level mosaics, in this case below 50%, their ongoing pregnancy rate is much reduced compared to the euploids. And once we get to a 1 chromosome mosaic, it's significantly reduced, as is 2 chromosomes. And then if you get to a complex mosaic, it's reduced again. So what we're saying is, if you want the highest chance of success, you want to counsel the patient to accordingly and know that you're truly transferring a euploid, then you really need to know that it is euploid. We can break it down further, and this is where the accuracy of the calling comes in, because a segmental change is not as bad as an entire chromosome. So if you have just a partial deletion, then the embryo generally does better than if it's a whole chromosome that's mosaic. And so not only does PGT-AI call whole chromosomes, it will also very accurately call these segmental changes. And again, this is increasingly important when we look at PGT-SR, which are by nature structural rearrangements, some of which can be very small. So you want to make sure that you're really assessing the data properly. So at a very top level, what we find is if you transfer a mosaic, then your ongoing implantation is much lower, your miscarriage rate is significantly higher, and your ongoing pregnancy per retrieval drops because of these. So we're not saying mosaics don't work at all, we're just saying it's very important to differentiate between the euploids and the mosaics. So we're now saying how important it is to look for true euploids. How do we find the true euploids? Well, we find the true euploids using our PGT-AI platform. So PGT-AI was developed and it was groundbreaking in that it used true big data and live birth outcome data. So the whole point about AI and computer learning is you need to train the algorithms on the truth. You use the truth to drive your learning, and this is where PGT-AI really comes in, because we have live birth outcome. So by comparing and learning off this data, we could maximize sensitivity and specificity during the development of the technology and then apply it to your PGT-A samples. So as I said, it is the only platform in the field built off live data outcomes, over 1,000 biopsy datas, where we know that that biopsy resulted in a live birth or a sustained pregnancy. We then validated that baseline from over 10,000 embryos. So we ran these 10,000 embryos, PGT-AI made an assessment and gave it a call. We then reviewed each of those calls and ensured that we could understand why that call was made. That doesn't mean to say we overrode that call. It may have differed from our initial subjective call. The fact is, did the algorithm make sense? Had it made a call which was understandable and believable, in which case, yes, it was. The accuracy is there. So by doing this, we have robust and accurate interpretation of our PGT-A. We've removed all the subjectivity. We're comparing euploids to euploids, and we're avoiding human errors with transcription. So to put that in perspective, what we used to do, and many others still do today, is you generate the data via NGS. So in our case, we used Illumina technology to generate data via NGS. That data is then visualized. So the data is plotted and the image is analyzed by people to try and understand if that data is different from normal, different from euploid. And that is subjective. Anybody who's done any kind of work where you've got multiple people visually comparing calls will tell you, and there's scientific studies showing that the mood of the person, the tiredness of the person, the training level of that person can change how they view data. And so that data, to counter that, we had the data looked at by 2 or 3 independent people, but still there is that subjectivity. And then based on that, the clinicians made a transfer on that human driven data set. PGT-AI still generates the data via NGS technology. We're still running Illumina technology, but instead of using humans, we've used all of that knowledge, all of that learning to build up machine learning. Now, every time a biopsy sample is set, it's analyzed exactly the same way, irrespective of which laboratory in the world, which person is looking at the data, doesn't matter. Every single sample is analyzed completely identically, and this avoids human subjectivity and avoids human errors. This is bringing the power of big data to the transfer decisions. So clearly technology is all very well, but what does it deliver? The whole point of innovating technology is to increase clinical accuracy and improve clinical outcomes. So what we found in our first few months of running this is that actually we were now reporting more euploid embryos. Now this is a reporting tool, it's a screening tool. We're not changing fundamentally the embryo itself. Humans are generally cautious, so if you are the person responsible for signing off a report and putting your name to the analysis, and you know there's a live birth at the end of that potentially that could be affected by a genetic disability, you are more likely to call something abnormal if you have doubts. By removing that subjectivity and by pushing it through a computer which doesn't suffer from such human emotions, we were able to more confidently report some euploids which may have previously been reported as mosaics. This increased not only the number of euploids we reported, but the number of patients with at least one clear euploid reported. And in fact, in this data set, it was an extra 1,300 embryos that were reported euploid. And they are spread out across the types. So as a relative increase, we went up 7.7% relative increase in the number of euploids being reported. There was a relative decrease of 4.2%, in aneuploid reporting, but also the level of the number of embryos we were reporting as mosaic decreased by 21.2% relative to the subjective calling. So it was a more robust calling, delivering more euploids back to the clinic. And the good news is they were spread over all the age ranges. So every single patient group benefited from this more robust calling, more scientific rationale, and a much stronger baseline to these calls. Obviously, the other key point is that transfers, you want as many transfers, not only of embryos, but as many patient couples coming through to have a transfer. And we found that when we looked at the data, not only were we getting more euploids, as I stated, we are getting more couples with at least one euploid, and it works out for about 1 in 25 extra couples were having a clear euploid transfer. So highly significant outcome for the patients and for the clinics. So this was a fantastic start, and we could have stopped there, but we didn't. We felt and we knew there was more we could do. So utilizing all the knowledge we gained from running PGT-AI1 and all the samples we've processed and the feedback we've received, we went for PGT-AI2. So this is the next generation of PGT-AI. So it's built on the same premise. So we have the embryo biopsy and live birth outcome data. But what we have now done is use much bigger sequences. So by using bigger sequences, we can generate more sequence data. By generating more sequence data, about 10 times more data than we used to produce, we now have more accurate sequencing. This is driven because we do paired end sequencing, which basically means that you get a much better alignment to the human genome, you get more accuracy where that sequencing lives, you get more data to analyze, and by having this extra data, we were able to see the SNPs within the data, so we have genome-wide or global SNP coverage. This is a world first for PGT-A. It's routine in prenatal testing and postnatal testing, but we're the first company to really bring it to PGT-A. And we have and this provides us with 2 independent methods for aneuploidy detection. Again, a world first for PGT-A. So the clinical benefits of the PGT-AI2 and the plus platform is that we have the most robust assessment for embryos in the market. So we have the two independent methods for confirming abnormality. So if we say it's euploid, we're doubly confident it's euploid. We can now detect all forms of ploidy. By default, male triploidy, which we've always been able to detect, but now it's a bit more robust, is there. So any embryos which are XYY or XXY, male triploidy, completely identified. If the parents are prepared to submit cheek swabs, we can also do the female haploid and female triploid. So just one copy of the female genome or 3 copies of female genome, that is available. As I say, it's an option. We need the parental cheek swabs. That's 2.0 plus. What we can also do is parent of origin of abnormalities. So again, if the parents are happy to submit cheek swabs, we can then start to analyze if the aneuploidy is coming mainly from mum, mainly from dad, a mixture of both, and start to look at how that aneuploidy is being driven in the embryos that we test. So SNPs are a very common form of genetic variation. The human genome is built up of approximately 3 billion base pairs, and each base is a nucleotide. And a polymorphism is where you have a variance. So it is potentially possible that within that 3 billion pairs of code that we all have within us, that one of those coding blocks could be different. And if it is relative to what we expect, then that is a SNP, a single nucleotide polymorphism. They result from natural variation. So each time a new generation is conceived, as the DNA is replicated, errors can occur over time, and that is just human evolution, and in fact it's evolution full stop. But it's a naturally occurring event. It's nothing to be either scared of or indeed concerned about. And most of these SNPs have no effect on health or development. They just live in our genomes and they make us rather than somebody else. So each individual has a unique pattern of SNP in their genome. So we've known for many years, and these have been utilized for many years as biological markers. So you inherit about 99.8% from your parents. So you'll get some from your mum and some from your dad. And these have been established methods for looking for unique parental allelic contributions. The other way people may have come across SNPs is through things like hereditary testing, online testing of population. Are you Caucasian? Are you more Asian Pacific? You know, SNPs are used for all sorts of genome testing these days. But for us, it allows us to see if that monosomy, where you only have one copy of a chromosome, has that one copy come from dad? If you have a trisomy, have you inherited 2 copies from mum and 1 from dad? It could be the other way around. But we can then look at those embryos and work out which partner is passing on their chromosomes or not passing on their chromosomes in the correct manner. Now, even without parental DNA, you expect to see a variation between 2 people when they create the embryos and create potential children. So what we have now is a gold standard NGS copy number, the same chromosome counting we've been doing for many years on NGS, looking at sequence reads. And that gives us our top plot, our just CMV top plot. Then what we have now is SNP counting, so we can count the number of SNPs and compare that to the expected number. And that is a completely independent test. So just to make it completely clear, this is the same biopsy sample. We amplify it, and in a single sequencing run, so a single assay, we get two different forms of measurement, the CMV and the SNP. And then we can compare those two answers, bring them together. And what that means is it removes some of the artifacts. So they should both agree. What it also allows us to do is be much more confident in small changes, segmental changes. And this is particularly important with PGT-SR, the segmental structural rearrangements, where quite often you get telomeric segmental changes to detect. But in terms of this presentation, it's about mosaic calling and double confirming whether it's euploid or mosaic. So our overall performance with this assay is that across all ages, patient demographics, etc., of those embryos that come in for testing, 44% of them are euploid. And 40% of them, roughly, are aneuploid. So the vast majority of your samples are either euploid or aneuploid. You have very clear understanding of the embryo. Your remaining 13% or so are split between these low-level mosaics and high-level mosaics. Now, if we think back to the beginning of this presentation, I showed how you get a difference in implantation potential between a euploid, a low-level mosaic, and a high-level mosaic. And so when you're counseling the patient and you want to say, look, this is a euploid embryo, statistically you've got this chance of success, you want to ensure that they are truly euploid. And this is where we are really driving this double confirmation of embryo status. Equally, if they are happy to have a mosaic embryo transfer and you want to try and give them some guidance on the likelihood of success, then knowing truly whether it's low level or high level mosaic is going to change that conversation. And so by driving all of this optimization of tests, we are providing that information back to the clinics to inform the patient's decision on which embryos and priority of transfer. So with PGT-AI 2, there are now two test options. PGT-AI 2, for all samples received after the 30th of March, will have PGT-AI 2 by standard. So this is an upgrade to our existing PGT-AI technology, so you get that increased accuracy of euploids and mosaics. And that is being offered no change in price to you. It's a service upgrade for our customers. What we also have, and which I touched on earlier, which is an optional extra, is PGT-AI 2 Plus. And what that gives you the option of having is the origin of aneuploidy. So when you see that chromosome monosomy or trisomy or a segmental change, has that aneuploidy been derived from a meiotic division in the oocyte or the sperm? We would also be able to say if that sample is female triploid, so copying of 3 copies of the female genome, and also there's 2 PN verifications, so we would be able to tell if that embryo contains 1 copy of mum and 1 copy of dad. The requirements for this is we do need parental cheek swabs. So this is a simple cheek brush kit sent out to the clinic ideally, or potentially the patient. They follow the instructions, they send us back the cheek swab kits. And we can then test for these. Ideally ahead of time, but they are available retrospectively. So if you do get some strange results and you want to dig a bit deeper, that is something that we can definitely do through a retrospective analysis through these parental cheek swabs. To be clear, that does not require any extra embryo biopsy samples. That's purely adding the cheek swabs, and then we do some additional analysis in-house of the samples we already have. Why that is important is that there is an assumption that the vast majority of whole chromosome aneuploidy are maternally derived, and we know from evidence that in 90% of the time that is. But what about the other 10%? What if you have a relatively young maternal age, but all the embryos come back as aneuploid? You wouldn't expect that. So is this a couple where more of it is derived from the paternal side? Again, meiotic segmental, so partial chromosome aneuploidy, is believed to be most often of paternal origin. But what if it isn't in this particular couple's case? So by doing the parent of origin of aneuploidy, we can enable the true assessment of where that aneuploidy is coming from, to enable, if they do want to go down a donor gamete decision, is it maternal or paternal, or potentially even both? So you get more confident donor gamete decisions. Equally, it may just reassure the patient that they know what's going on. They can either keep trying or they can go away and think about it, but they have that peace of mind. They understand a bit more about what is going on with their embryos. And so, just as a kind of top level overview of the service that we will then be offering, is everybody gets PGT-AI 2 by default. So your 2 independent assessments of ploidy, and that is driven because we're just generating far more data and allowing a deeper analysis. The option is this PGTi Plus in the red box, which allows all the detection of female triploidy, parent of origin testing of meiotic changes, and the 2PM verification. As I say, you do need the parental cheek swabs to be able to do that. And so, in summary, PGT-AI is there to deliver the best clinical impact for the clinics and the patients. That is its design, that is its rationale. And because we've improved the accuracy and we've removed subjectivity and we're avoiding human errors, we're definitely well on the path to this. We've now added in this secondary assessment of abnormality. We're now even more confident that when we say it's euploid, it's euploid. This gives increased confidence when you're doing an embryo transfer. We know that a true euploid has a lower miscarriage risk compared to a mosaic embryo. So again, a positive message. And so when transferring euploids, which are truly euploid, then we should see a positive impact on pregnancy rates and continuing and ongoing pregnancies and live birth. So that is where we are with PGT-AI today. That's not to say that we're not continuing to innovate. Of course we are, and you will see more updates to the PGTi platform from Cooper over time. But I just want to reiterate that today we have this PGT-AI 2 with a secondary assessment to enable you to have the highest clinical impact and the highest success rates of embryo transfer. So I thank you for your time, and if you do have any questions, please feel free to email us, either me directly at david.crimes@coopergenomics.com or global support at coopergenomics.com, and we will get back to you with all your answers. So once again, thank you for your time, and hopefully speak to you soon. Thank you.